Hello, my name is Nadeau. I'm a master's student at the University of Ottawa, and today I will present to you a protein-protein interaction network guided functional enrichment analysis for the identification of dysregulated pathways in different breast cancer subtypes. The focus of this study is to further characterize the proteome. The proteome is the ensemble of proteins in a given environment. Proteins will often interact together in order to accomplish their functions, and therefore affect the biological processes that occur in this environment. To further characterize quantitative proteomics data sets, we will often rely on functional enrichment analyses. These analyses will look for overrepresented functions in a set of proteins of interest. This method often relies on arbitrary cutoffs and can therefore disregard a lot of useful information. Geneset enrichment analyses, or GSEAs, have been shown to address this problem by determining if biological processes are non-uniformly distributed in a ranked ordered list based on differential expression of the proteins. In addition, the use of external data such as protein-protein interaction networks can help capture useful information in a functional enrichment analyses. For example, this GoNet study identified locally overexpressed pathways in a protein-protein interaction network regardless of protein differential expression. My method uses the BioGrid protein-protein interaction network as the framework for the functional enrichment analysis. We can integrate protein expression as a measure of fold change between two conditions of interest, and then weight interactions such that two proteins that have similar differential expression will seem to cluster within the network. Finally, we can annotate the network using gene ontology information and identify pathways whose proteins are both differentially expressed and clustered in the network. For a given Go term, we will assess the clustering of its proteins using the total pairwise weighted distance, and then we can assess the significance of this clustering against a null model. We applied this method to the study of breast cancer subtypes. We used the data from the Nadeau et al. Study, which compared the HER2 positive and triple negative breast cancer tumors. Here we use a false discovery rate to assess the clustering confidence of our algorithm against a set of randomized Go annotations. And what we see is that we're able to identify a large number of significantly clustered Go annotations at a very low false discovery rate. So here, a false discovery rate of 1%, we identified approximately 3,000 Go terms in both a HER2 positive, triple negative weighted network and in an unweighted network. We looked at the overlap between the HER2 positive, triple negative weighted network against the unweighted network, and what we see is that there is over 2,000 Go terms that are shared amongst these two approaches, and these are most likely due to the innate clustering that occurs in the BioGrid protein-protein interaction network. And if we were to filter the results, we get 168 unique Go terms to the HER2 positive and triple negative breast cancer subtypes. Looking deeper into these significantly dysregulated biological processes, we see things like COP2 vesicle coating, negative regulation of sprouting angiogenesis, response to insulin, that are all biological processes known to be affected in breast cancer subtypes. Finally, we look to benchmark our approach against the Markov clustering and ontologizer method. So Markov clustering enables the identification of clusters in a given network, and the ontologizer provides functional enrichment of these clusters. And what we see when comparing at similar thresholds is that we do have some overlap in the significant Go terms, but our approach is able to identify more information that would not have normally been identified in a standard approach. To conclude, our method enables the identification of dysregulated pathways through the identification of significantly clustered proteins in a protein-protein interaction network. This method will be useful for the characterization of many quantitative proteomics data sets, and our algorithm can be used on both weighted and unweighted networks. I would like to acknowledge Mathieu Lavallée-Adam, my supervisor, as well as my lab mates, in particular Anastasia Bivsheva, who did some work on this project, and my funding agencies. Thank you very much for listening.